Hello everyone. Today we'll discuss the part 14th of the important notes for biochemistry so that you can easily able to answer the one-liners and the MCQs in your exam. Trinucleotide repeat expansion. Now over here, a sequence of 3 bases that is repeated in the tandem will become amplified in number so that too many copies of the triplet occur. What happens over here, as you can see, a triplet, or over here this is the triplet, right? Trinucleotide, over here this is the trinucleotide, right? Now what will happen? There will be expansion of this particular trinucleotide will actually occur in the repeated manner. Okay, so that what happens? Too many copies of the triplet occur. Now disease due to the trinucleotide repeat expansion is the Huntington disease, Fragile X syndrome, frame shift mutation, splice site mutation. Now first of all, the Huntington disease. Now this Huntington disease is actually a hereditary disease and a neurodegenerative disease. Now over here what happens is trinucleotide repeat expansion occurs of the codon for the glutamine, that means CAG basically repeats over here. Okay, and this leads to the formation of an abnormal protein which is degraded easily, producing toxic fragments that aggregate in the neurons. Okay. Now what is Fragile X syndrome? Over here the trinucleotide repeat expansion basically leads to the hypermethylation which causes the gene silencing. Now over here the Fragile X syndrome, what happens is there is mutation of the Fragile X mental retardation 1 or the FMR1 gene mutation basically occurring, and this particular gene is present on the X chromosome. Most commonly, an increase in the number of CGG trinucleotide repeats in the 5' untranslated region of the FMR1 basically occurs, causing the Fragile X syndrome. So just remember over here, the main trinucleotide repeat occurring in the Fragile X syndrome is the CGG trinucleotide basically occurring at the FMR1 gene, causing the mutation. Frame shift mutation. Now over here, addition or the deletion of the nucleotide which is not divisible by 3 alter the reading frame. Splice site mutation. Now this can interfere in the removal of the introns from the primary transcript. Now large scale mutation or the macro alteration, or we can say chromosomal mutation. Now this basically includes amplification or duplication, deletions, inversions, translocation and the aneuploidy. Now an inversion that involves the chromosome's constriction part, that is the centromere part. We discussed about this before also, that centromere is the constricted part of the chromosome, right? So an inversion at the chromosome's constriction part or the point or the centromere is known as pericentric inversion. An inversion which occurs in the long arm. Okay, this is the long arm. Okay, this is short arm. Long arm is considered as Q, is given abbreviation as Q, where the short arm is given abbreviation as small p. So an inversion which occurs in the long arm or the short arm, that means P or Q, and does not involve the centromere is called a paracentric inversion. So inversion involving the centromere, is known as a pericentric inversion, while the inversion involving the arms, either short or long arm, and doesn't involve the centromere, remember, the centromere is known as the paracentric inversion. Translocation occurs when a piece of one chromosome breaks off and attaches to the another chromosome, and it can be balanced or unbalanced. It can be normal or balanced or unbalanced. If no genetic material is lost or gained from the chromosomes, then it is known as the balanced translocation. Okay, and this occurs between the homologous chromosomes. If there is a gain or loss of the genetic material from the chromosome, then it is known as the unbalanced translocation. Now, Robertsonian translocation. Now, in the Robertsonian translocation, there is joining of the long arm of 2 chromosomes. Okay, and the tiny arms, or we can say the tiny short arms, are usually lost. So as you can see over here, the long arm of these two are actually combined together. Aneuploidy is abnormal chromosome number It can be trisomy, means 3 chromosomes instead of 2, or it can be monosomy, 1 chromosome instead of 2. So as you can see over here, there are 3 chromosomes instead of 2 at 21st position in the Down syndrome known as the trisomy 21. So today up till here, we'll discuss about the further important notes in our next video. Thank you so much.